Good afternoon, everybody, and welcome to the first Retina Australia webinar for 2023, the latest in research from Retina Australia and grant recipients. My name is Sally Turnbull, and I'm the Administrative Officer for Retina Australia, and I'll be the facilitator of this webinar today. So first of all, I'd like to introduce our CEO, Julia Hall, to say a few words of introduction. Thanks, Julia.
Thank you, Sally, and good afternoon, and thank you all for attending our research webinar today. As Sally mentioned, for those who don't know me, my name is Julia Hall and I'm the CEO of Retina Australia. Before we get started, I'd like to begin by acknowledging the traditional custodians of the land on which we meet today. I'd also like to pay my respects to elders past and present. We're very excited to have Associate Professors Penelope Allen and Lauren Ayton with us to provide updates on their inherited retinal disease-related research. Thank you both for taking the time to present today. I'll now pass back on to Sally to introduce our guest speakers in more detail.
Thanks, Julia. So our first speaker this afternoon is Associate Professor Penny Allen from the Centre for Eye Research here in Melbourne. Associate Professor Allen graduated from the University of Melbourne in medicine and specialised in ophthalmology. She then subspecialised in vitreoretinal surgery. She is a research fellow at the Centre for Eye Research Australia with a clinical appointment at the Royal Victorian Eye and Ear Hospital as head of the vitreoretinal surgical unit. She was a principal investigator on the Australian Research Council through its special research initiative in Bionic Vision Science and Technology grant to Bionic Vision Australia. She is the co-recipient of an NH and MRC Excellence Award. Associate Professor Allen was key to the developing the surgical technique for the suprachoroidal retinal prosthesis, resulting in the first clinical trial of this device in 3 human patients. For this, she was shortlisted for an Australian Eureka Prize for interdisciplinary research. She has subsequently led a second trial of a fully implantable device with 4 patients. She has presented over 60 conference posters and talks and has published more than 90 peer-reviewed journal articles. This afternoon, Penny is going to speak on improving real-world mobility and assessing long-term safety outcomes with a retinal prosthesis or bionic eye. Thank you very much, Penny.
Thanks, Sally, for that lovely introduction. I'm just going to share my screen. And it really is a great honour to be able to speak to you all today. It's wonderful to have the support of Retina Australia, and it's really a great privilege to be able to talk to you about the work that we have done over the last few years, but then also in particular talk about the work that has been supported by the funding from Retina Australia most recently. So just to introduce things, as Sally mentioned, we ran a prototype clinical study of our suprachoroidal retinal prosthesis during 2012. And we recruited 3 patients for this trial, all of whom had end-stage retinal dystrophy and bare light perception vision. And then in 2018, we recruited 4 more patients for our second generation trial. Once again, all these patients had retinal dystrophy and bare light perception vision. And you can see on the model on the right here, the externals that we used for that particular trial. So when we say we developed a bionic eye or retinal prosthesis, what do we really mean? Well, what we developed, and this was in conjunction with the engineers from the Bionics Institute, was a silicon array with platinum electrodes that you can see here, that was placed within the suprachoroidal space, and it had a lead to a percutaneous connector. The reason that this initial device had a connector was so that we could vary the levels of stimulation greatly, because no device had been placed within the suprachoroidal space previously, and we wanted greater flexibility of stimulation. But it did mean that this device was designed to be used within the lab. It was the results of that work, however, that led us to our second generation device with an increased number of electrodes and a wider field of view, and two receiver stimulator units that were implanted beneath the skin. So this is a fully implantable device which was designed to be used at home. So over the time period of these two trials, we performed 7 surgeries, and we used the same ocular procedure for both devices. In all 7 surgeries, the device was able to be positioned safely with no intraoperative complications, and it was placed beneath the macula in all cases. At the end of the surgery, the impedances of the electrodes were measured, and there was no loss of functionality. And you can see an image here of one of our 44-channel patients with the device in situ. There's a colour photograph and also a red-free photograph. So during the follow-up, of course, we've been following the patients from the 24-channel study for 10 years. One of the 24-channel patients did need to have his device removed due to an unrelated health issue. And the 44-channel patients have been followed for over 4 years. And that follow-up consists of clinical assessments, colour fundus photographs, and OCT used to assess the stability of the devices and also assess retinal health. And we also assess the position and stability of the device within the eye by measuring the distance from the leading edge to the optic disc and comparing rotation and translation to baseline. And in the images here, you can see the colour fundus photograph of a device in situ with the characteristic features of retinitis pigmentosa. And then below, an image of the OCT that's been used to measure the position of the device. And one of the things that we measure is retinal thickness. We know that with time, patients with retinal dystrophy have progressive thinning of the retina, but we wanted to be able to demonstrate that the devices did not alter the natural history of this disease. And we can see the plots on the left of the retinal thickness from the participants with the 44-channel device, and there is just overall a slow thinning of the retina consistent with retinal dystrophy. And when we look at electrode to retina distance, we were concerned that in the first trial that fibrosis developed around the device. And so we've conducted measurements within the second generation trial also, and demonstrated that certainly there is some fibrosis that does develop around the device, but this is stable in the 44-channel patients. And the OCT, once again, is used to measure the distance between the electrode and the retina to document the development of any fibrosis. We also constantly monitor the functionality of the devices, and it was very pleasing that during the 24-channel trial, all of the devices remained completely functional with no loss of electrodes during the 2-year study. And during the 44-channel trial, only 4 electrodes overall became open circuit, that is non-functional. We believe that the stability of the device in the suprachoroidal space contributes to this low loss of functionality in the device. So what about the follow-up of the patients and assessment of the functionality of the devices? Well, we call this work psychophysics, and it initially consists of testing the thresholds of every electrode in the lab and generating a map of those thresholds, and also the phosphene, that is the visual perception that the patients have from the stimulation. We then use a number of tests within the lab to assess the functionality of the devices. And we can see on the left, one of our patients assessing using the moving bar test, where we have a moving bar on a computer screen in one of 4 directions. We also use square localization on a computer screen as well. And then we move on to more useful testing, really from the point of view of patient use of the device, what we call O&M, orientation and mobility work. So identification of a door, and you can see one of the patients above, testing a high contrast door in our testing room, and also obstacle course. We can see one of our other patients on the right participating in a tabletop task, and that is locating an object, be it a cup, a placemat, a bowl, or cutlery, in a position on the table, and also attempting to identify that. And we were able to show with our original vision processing algorithm, that is, we call a high contrast algorithm, that we could show that the device on function was better than device off for tabletop search and door detection. And you can see some video of one of our patients demonstrating door detection on the right. And this is, once again, our original high contrast vision processing algorithm with one of our participants sorting washing. And we've videoed the screen of the computer on the right here to demonstrate the phosphenes that are generated by the patient using the device to assess the items of clothing on the table as background. And with this high contrast vision processing algorithm, then the accuracy of sorting washing is excellent, and other tasks such as obstacle course are also able to be completed. And this is another one of our patients using the original vision processing algorithm within a shopping mall. And we've got the video on the left. He's using this in addition to the cane. We're attempting to demonstrate the phosphenes in the video on the right. And you can see as he comes closer to an object, then there is stimulation. And to move forward, then moving in an area where there are no phosphenes is an area that is free and able and clear. However, our overall aim has been to improve the functionality of these devices. And so to that aim, we developed a new depth algorithm that would enable the patients to assess proximity of obstacles in front of them, which, as they come closer to the obstacle, generates a brighter phosphene. And to that aim, we can see the new headset on the model on the right. And this headset has 2 cameras to enable depth perception and a new vision processing unit with the ability to be able to swap between the algorithms. And so here we are assessing the 2 algorithms against each other. And it is this work that our Retina Australia funding was so vital for. So we compared our original high contrast vision processing algorithm with the depth algorithm. And this is one of our participants in the obstacle course. In this setting, in a clinical trial, then we randomly run trials of the obstacle course. The obstacles are all randomly placed within the course, and the patient doesn't know which vision processing algorithm they're using at a particular time. And you can see with this overhanging object here, our participant is able to identify that there is something hanging overhead and move around that. And then he comes upon the mannequin and once again is able to identify that the mannequin is in the obstacle course. As an additional part of this assessment and comparing these 2 vision processing algorithms, then we conducted outdoor street work. And you can see here, our patient was able to see the young mum with the stroller and pause before walking on. And I'd like to emphasise that it is this outdoor street work which is completely novel and has not been actually performed by any of the other Bionic Eye groups. And there's 2 clinical trials of obstacle avoidance and streetscape work are vital for us to get regulatory approval and evidence of functionality of these devices for commercialisation at a later date. So what can I say about the results of our obstacle avoidance task? Well, overall, the depth algorithm performed better than the contrast algorithm for object detection. For low contrast objects, in particular, the depth algorithm was better. Our original high contrast algorithm performed well for high contrast objects, but the depth algorithm was better for mannequins, overhanging boxes, and large bins. And it was the contrast algorithm that was better for ground-based boxes. So that as an overall algorithm, then the depth outperformed our original algorithm. In the outdoor streetscape work, we were able to demonstrate that the highest proportion of objects was detected by the device, 70.3%, as opposed to cane, 22.3%, or body, 3.8%. And of course, without the device, then our participants would be using cane or body. Correct identification was equivalent between the 2 algorithms, and they were able to correctly identify the objects in a third of the time. One of the problems that we have with our high contrast algorithm is that shadows are often incorrectly identified as objects, and the depth algorithm is very useful to address this mistake. So overall, our participants felt that the device added a degree of confidence to their navigation during the outdoor streetscape and obstacle course work. So what can we conclude after this recent work? Well, our surgical procedure is safe and reproducible, and we're able to position the device beneath the macula reliably. The devices are stable after an initial settling period, and they remain functional, and we believe that's due to the stability of the devices within the suprachoroidal space. But we're excited that our new vision processing algorithm can add functionality and aid navigation and activities of daily living for the participants. And our participants like to be able to use the different algorithms and swap between them during use. So once again, I'd like to thank Retina Australia for funding this recent work. I'd like to acknowledge, obviously, our other funding that we have had over the years to participate, to contribute to this work. Our most important acknowledgement goes to our patients, however, who without their dedication and hard work, we would not be able to progress this work. None of this happens without a large team, and my team at the Centre for Eye Research Australia are a great support, and we've had fantastic collaboration with the Bionics Institute, the University of Melbourne and the Royal Victorian Eye and Ear Hospital. We've also need to thank Professor Nick Barnes from the Australian National University, who is the developer of these vision processing algorithms. Thank you for your interest.
Thank you so much for that, Penny. That was fantastic. So exciting to see these amazing developments. So we'll move on to our next speaker, and we'll do questions at the end. Our second speaker this afternoon is Associate Professor Lauren Ayton, also from the Centre for Eye Research and the University of Melbourne. Associate Professor Lauren Ayton is the head of the Vision Optimisation Unit and a Principal Research Fellow at the Centre for Eye Research. With Dr. Tom Edwards, Lauren co-leads the Venture Registry, which is aiming to collect clinical and genetic information on people living with inherited retinal disease. She's an investigator on several industry-sponsored clinical trials and also runs a number of investigator-initiated research studies. This afternoon, she will provide us with some updates on gene therapy for inherited retinal diseases. Thanks so much. Lauren.
Great, thank you, Sally, and thank you to Penny as well for a fantastic starting talk, as she always gives. So again, as Penny said, it's a real pleasure to be able to share some of the research that we're doing at CIRA and at the University of Melbourne. And I guess what I really wanted to do is almost take a little bit of a step back from what Penny was talking about and talk about how we are inviting people to be involved in our research. So this is talking about the Venture Registry, which is our inherited retinal disease registry for people to get involved more in this sort of research. So that will be sort of the overview of my talk. I'm going to talk about how we find people, how you can get involved, and also give you an update on some of the clinical studies we're running, as well as talking a little bit about some Retina Australia funded work looking at people's perspectives on these emerging treatments. So I just have some disclosures. So I am an investigator for a number of trials and consultant for some companies as well. So the Venture Study team is really led by myself and Dr. Tom Edwards, who many of you on the call will know. So Tom is a vitreoretinal surgeon that works in Penny's unit at the hospital. And our team is really, spearheaded by Dr. Cece Britton-Jones and Mr. Parker Trung. Sashini, unfortunately, has just left us to go back to Perth recently. But the idea with our registry is that we are trying to learn more about inherited retinal diseases in general. So we are bringing people in for these assessments, which is looking at the clinical presentations of inherited retinal diseases. So that's measuring people's vision, taking pictures of the eye and scans, as well as asking questionnaires about how people feel that their inherited retinal diseases affect their life. The other aspect of this registry, very importantly, is genetic testing. And I'm sure it's no shock to anyone on this webinar that genetic testing is one of the most important tools that we now have in IRD management. because we can identify people for these upcoming treatments from their genes. Once we have people on the registry, it gives us a really powerful tool to then recruit for our clinical trials. And as Sally mentioned, we're running at the moment 7 clinical trials in this space, with more, on our doorstep as we speak. And I'll talk about these a bit more in detail later. But really importantly, what Venture also allows us to do is tie back into the preclinical work. So this is the basic science in the lab, where Tom and the team are coming up with new treatments. And we work really closely with collaborators across different units within CERA and the university. And it might be things as simple as giving some input from a clinical point of view as to what could be important in a treatment, all the way through to sometimes taking samples from people who are involved in our research in the Venture registry, and then using those samples in the lab to develop new treatments. So it's quite a nice sort of circle of life. Now, we've recently published on the first 150 people that are on the registry, and we're now up to almost 300 people, which is fantastic. And a huge thank you to everyone on the call who has registered already. And what we're finding so far is pretty similar to what we see overseas. So the most common inherited retinal disease that we see is retinitis pigmentosa. We have a number of people with Usher syndrome, and many of you will be aware that Usher syndrome is the leading cause of deaf blindness. Part of the reason that we do have quite a high proportion of people with Usher on our database is that we do run clinical trials in this space. And so we'll talk some more about that later. But overall, we get pretty similar breakdowns of different subtypes of IRDs that you see in the US and in Europe. And really importantly for us, this data is actually very powerful for us to then talk to industry partners and companies that are developing treatments and say, okay, these are the really common inherited retinal diseases that we see, and this is where you need to target your R&D. So again, another reason to get involved if you're interested. Another thing that I often get asked is, is my vision too bad to get involved in this registry? And the answer is no. So as Penny just mentioned, CERA and Penny herself are leading trials into things like the Bionic Eye. And there's also new treatments coming out that may help people with end-stage vision loss. And the other aspect of our research program is really looking at low vision aids more generally. So mobility and electronic travel aids and things like that. So we really want to hear from everyone. And we do have everyone on our registry, from early vision loss all the way through to what we call ultra low vision. And this is really important because there are treatments being developed for the different stages of inherited retinal disease. So as we'd all be aware, these are degenerative conditions. And so we can sometimes detect disease at a very early stage while the vision is still normal and we have very limited clinical signs. And obviously that can go through to being quite a pronounced loss of vision. And the really exciting thing for all of us really is that there are therapies being developed for every stage of these conditions. So gene editing and gene therapies are more targeted towards early disease and early vision loss, through to things like stem cells, optogenetics, and vision prostheses, which are more for the end stages of vision loss. And so this obviously brings a lot of hope to both the researchers and people living with these conditions. And of course, it all comes back to Luxturna. This is the poster child of our field at the moment. And again, I'm sure pretty much all of you will be aware of Luxturna. So this is a gene therapy, a gene replacement treatment for a form of Leber congenital amaurosis that's caused by a gene called RP65. And so it's a once-off treatment. It's an injection underneath the retina that gives the correct version of the gene. And so this has really been a game changer in our field. So now we have a regulatory approved gene therapy, which is being given to people in Australia now as we speak. And this has opened up the door for the next generation of treatments. So what's coming? So this is from a recent review that our team did. And what I've got up on the slide is a graph showing how many times a particular gene has been in the literature. So whether that be scientific papers or conference presentations or listing on clinical trials websites. And you wouldn't be surprised that RP65, the gene that has the treatment in Luxturna, is the most commonly researched gene to date. However, there's some really close candidates following closely behind, and they are Leber hereditary optic neuropathy, choroideremia, X-linked retinitis pigmentosa that's caused by a gene called RPGR, and Stargardt disease. And so these conditions have all got clinical trials underway at the moment. And the other genes that are listed, and I can give people a verbal rundown of them if they would like later. There's a few, but there are treatments underway being developed for a whole range of different genes. So what we're really interested in is finding people that can benefit from these treatments. And so a really interesting thing is finding out what genes people have. And so we recently did a survey of the literature where we looked at where the genetic diagnoses have been published. And unfortunately, there's not a huge amount of data from Australia. And so that's something that we are working towards changing with places like the Australian Inherited Retinal Disease Register, which is funded by Retina Australia. We're trying to collect more and more national data so that we can get a better understanding of what types of inherited retinal diseases we have in Australia. But as expected, places like the US and China do this very well already. And the change really has been over the last decade in terms of the accessibility of genetic testing. So a decade ago, it was very expensive. It was very hard to get genetic testing. And when you did do testing, quite often you didn't get a result. And so back, for example, in 2011, only about a quarter of the people that had a genetic test actually got an answer. Thankfully, that's much better now. So we're up now around the 60 to 70% success rate when we do a genetic test. And we also have a lot more advanced techniques now to try and learn more. So we can do things like look at the entire genome, not just one specific gene at a time. And this is really accelerating the field and helping us learn more about what types of genes cause what types of differences in vision and in the retina, as well as obviously leading to new treatments as well. So a really interesting and important part of all of this work is actually finding out from people that have these conditions what they actually want. So if there are treatments available, would they be interested? What are the barriers? Those sorts of questions. And so we were very fortunate to receive some funding from Retina Australia recently for a grant that was headed by Associate Professor Heather Mack from Sierra. And what we looked at is the attitudes to gene therapy for the eye. So with a multidisciplinary group, we had a whole range of people involved in developing the survey. We came up with an online survey that we then distributed through places like Retina Australia, and we had almost 700 people reply. And this is absolutely mind-blowing for this type of research. So we think it's about 5% of people that have an IRD in Australia replied. So 1 in 20, which is really amazing for this sort of work. So a huge thank you to those of you on the webinar who helped us out. So interestingly, what we found is that the mean age of the people that had an inherited retinal disease was 54. And we had people replying between the ages of 18 and 93. And we also had 42 people who were caregivers reply to the survey, which was great. The really interesting finding for us in terms of our quest for treatments was that most people would take up gene therapy if it was available for them. So it was 92% of people that responded said that they would have gene therapy if they were able to. But one of the challenges is that only 28% of the people that replied also said that they thought they had a good idea of what that entailed. And so it's a little bit challenging, obviously, to know whether or not you would have a treatment if you don't really have good knowledge of that treatment itself. So this is highlighting a need for us as clinicians and as researchers to improve our education in this space. And so we're going to continue to work with Retina Australia to make sure that we get the information that we are learning out to your members as quickly as possible. There were some other really interesting findings from the survey that we did. So we looked at whether people had had previous treatments before, and we found that most people hadn't done anything. There were some people that had tried vitamin A or herbal remedies, and acupuncture was the third most common other treatment that had been trialled. In terms of barriers and reasons that people wouldn't go ahead and get gene therapy, most people said that they didn't really see any big issues, but the ones that were highlighted were things like cost, potential side effects, and also the fact that these are still early stage treatments. And so we cannot be sure that it's going to work for an individual. And that's a really interesting and key thing for people to keep in mind. While we're really excited about these treatments that are emerging, it is still early days. And so you need to be aware of the risk-benefit analysis before you make a decision to be involved. So this was absolutely, like I said, an outstanding response to this survey, and it really was because of the community involvement. So mainly to Retina Australia, to be honest, both for the funding and also the fantastic dissemination of this survey to your members. So thank you. And I also want to acknowledge Vision Australia, Blind Citizens Australia, Usher Kids Australia, Cure Blindness Australia, and the Retinal Disease Registry in Perth. So turning over now to where we stand in terms of clinical trials. So I have some information up on the screen. If you can't see this, again, I'm very happy to provide this in another format later. But basically what it's highlighting is that we have active clinical trials in Leber congenital amaurosis, Stargardt, Usher syndrome, and also in macular degeneration. So we are a treatment site for Luxturna for patients that have the RP65 mutation. And so that is actually a post-market treatment. So it's a commercially available treatment that you can have through the eye in your hospital. We're running earlier clinical trials for oral medications for both Stargardt's disease, and that's for adolescents, so for 12 to 20-year-olds, and also for Usher syndrome as well. And we do have a gene therapy trial underway for macular degeneration. The take-home message that I'd really like to stress to everyone listening today is all of these clinical trials and all of our research comes back to the Venture Registry. So this is really the centre point of our research program. So we are looking for anyone who has an inherited retinal disease that thinks they might be interested in research, whether that be interventional, like getting a drug trial, or whether it be looking at things like low vision aids or just surveys about how your vision has been affected. Venture is our linchpin for all of that. So please, if you're interested, do contact my team and I'll give you those details in a minute. In terms of our specific investigator-initiated research, so this is research that we're doing not with support from industry. We've got a number of projects underway at the moment. And again, I would pretty much say that there's something for everyone in terms of whether you can get involved in research. We're particularly interested in people that would like to share their experiences of early vision loss. And this has come from some discussions that Cece Britton-Jones, our postdoc fellow, had with some people that were involved in our research, of saying that there's some great resources available for when you need low vision aids and when you need to access, NDIS or those sorts of things. But potentially in the earlier stages of vision loss, we don't have as many good supports available. So this is a study that's underway with interviews. So it's done via Zoom or the phone, where people can really talk about their experiences of early vision loss and what we could do better as a community to help in that stage. We are very interested in as well in female carriers of X-linked IRDs and of choroideremia. And this is a really interesting space because historically, females that had, affected males in their family were told that they were at very low risk of losing vision because they only held one incorrect copy of the gene rather than the two that males do. What we're finding, though, is that some female carriers can be very affected, and unfortunately, some women can actually lose vision. And so we're trying to learn more about that. So why is it that some women lose vision and some go through life with their vision, unaffected? And so I have a PhD student, Senna, who is working on this. And so again, if this sounds like you or someone you know, please do reach out. And the final study that we're actively recruiting for and really looking for volunteers for is people who are legally blind from IRDs who'd be interested in trialling some new sensory substitution mobility aids. So these are things like audio cues and vibrating devices that can help you identify when there's obstacles in the way. And we're going to run some assessments within a physiotherapy lab that's got the biomarker sensors on the body, kind of similar to what they do for animated movies and things. And so we will be using that very advanced technology to look at how we can help people with their mobility. The last thing I just wanted to finish up by saying before we go to questions is that we at CERA and the university have just become a site for the Foundation Fighting Blindness, which is a really exciting advance for us. So FFB are one of the largest philanthropic groups in the IRD space around the world. And there's a large global consortium which has pretty much all of the leading institutes in the US and Europe involved. And we've now become the first Australian site for that as well. And what that means for you guys is that we will now have access to earlier trials for new drug treatments, but also natural history studies. So we will be able to be involved in collecting data from Australia to share with our colleagues overseas so that we can get bigger numbers in our studies, get treatments through more quickly, and hopefully get some more options on the table. So I'm going to finish up there just really by saying, again, a huge thank you to Retina Australia. So our work is very much reliant on the wonderful work that you do, not only in financial support, but probably more importantly for us in connecting us with your community. So I've got a photo of our lab team. We're growing rapidly. So every time I do a meeting, I feel like we add in another 5 people. But that's very exciting. It's because we're getting a whole lot more research happening. So if you're interested in anything that we've talked about today, there is an email address. It's ird@groups.unimel.edu.au. And again, we can send this email address out later. And probably the other thing I'd emphasise is just to be aware that, we all do work very closely at CERA as well. So, if you did want to get in touch with someone like Penny, we can all, be contacted fairly easily. So we work well together. So I'd like to finish off there and thank you for all listening today and pass back to Sally, and hopefully we have some time for questions.
Great. Thank you so much, Lauren. And Penny, that was tremendous, and it's so exciting to hear about all of these new studies that are taking place and to think about the possibility of people being involved in them. It's great, isn't it? So we do have time for some questions, and so if you'd like to ask a question, just type it into the Q&A section, or you can raise your hand, use the raise your hand option at the bottom of your screen, and we will respond to those in the order that they appear. When we say your name, we will enable your microphone, and you will need to unmute yourself so that you can ask your question. And then after your question has been answered, we'll turn off your microphone again. In order to raise your hand, you can just click on the bottom of the screen, or you can use Alt-Y on your keyboard. If you've joined us on the phone, you need to press *9. And to unmute your microphone, you can select the unmute button on your screen or just press the space bar. If you've joined on your phone, you need to press *6. Alternatively, you can type your question in the Q&A section, and I can see that there are a few there. So somebody has said, thank you both. I had several questions relating to Stargardt, but you have answered them all. Bravo to you both. So that's good. Cameron Carter had a question. Why can't the algorithms be combined And or is that something for the future? What is the future like in relation to AI coming on board as well? I'm guessing that's a question for Penny.
Yes. Unfortunately, I think you've switched my video off, Sally, but I can certainly answer that. The algorithms are used as alternatives in the vision processing unit, but we have in fact developed further algorithms which we are currently testing as well. And as far as AI, then that is also the next thing that we have planned for these devices. And I think the really exciting thing about this is that we can, alter the externals. That is the glasses, alter the cameras, we can alter the vision processing unit, but we don't need to do anything about the implanted retinal prosthesis. We can improve the functionality without actually doing further surgery.
That's great.
Absolutely. I might just jump in as well, Sally, and just add, because artificial intelligence is obviously a really important and interesting area for all of us. The other aspect where I think it's going to be really important for inherited retinal diseases is in terms of image analysis. So one of the challenges in IRDs is that each single subtype of condition is very rare. And so we're actually now talking to places like Moorfields Eye Hospital in the UK and some places in the US about whether we could combine images. And this gets tricky because obviously everyone has to consent to this and we need to do it in a very secure way. But there's a potential that you could use artificial intelligence to look through a large number of images and identify new signs of the disease. So we might be able to detect something that then becomes a diagnostic tool in the future. So again, very early days for that because we need to really work through how that works, sharing that sort of data across sites, but could be very huge.
It's a bit like that thing where, we don't know what we don't know. It's going to take a while to work out what opportunities there are.
Absolutely, absolutely. And unfortunately, when it comes to artificial intelligence, the computers are actually a lot better at us than looking at images at that scale. So in an individual image, obviously we're very good at seeing what's there, but looking for patterns across thousands of people, that's where AI will come in.
Fantastic. So we've got another question from somebody. Can you please confirm if genetic testing is part of the process when joining the venture registry? Will a DNA sample be taken and sent for testing?
So that's definitely one of our key things that we're trying to give access to everyone that we can. It's changed slightly. So there basically has been ups and downs in terms of how much genetic testing costs. So some of the companies, Invitae and Blueprint, have had what we call sponsored genetic testing programs where we don't have to pay for the costs of the sequencing ourselves. So we went through a phase where absolutely everyone that joined the registry had genetic testing. So we were sending out kits to people at their homes. The system has changed slightly now. And so now what we're doing is registering everyone that's interested. And then when people come in for their clinical visit, we then do the genetic testing at the same time. And this works a lot more effectively because when we send off your results to be looked at, they actually also need to know the clinical data to be able to get a good diagnosis. So we really need to be able to give them information about what the back of the eye looks like, about what the vision is and things that we need to see you for. So very long answer there, but basically there are two options with Venture. If you can't come in to see it, we can put you on the registry still. We can collect all the information that we can on the phone. And that means you're still then able to be involved in trials as they come up. But our preference is if you can come in to see us in Melbourne, then we can also do the imaging and the genetic testing as well for you.
That's great. Does anybody else have any more questions? I have a couple I can ask. I've got quite a few actually. I was going to ask Penny, just in relation to the issue that you said that your product is the first one that works outside, sort of in the general, just in a general sort of environment. Is that to do with the bright light that's outside, or is it just because it's a more complex environment?
No, it's not quite that our device is the only one that works outside, but we've specifically done streetscape testing of these different algorithms. And that's not something that other Bionic Eye groups have done. But interestingly, of course, at the moment, there are not options for a commercial Bionic Eye for a patient with retinal dystrophy. There is a second site from the United States who did have a device, are not producing their device anymore and not further implanting it. Pixium in France, who had a device for patients with RP also, and Retina AG in Germany have all stopped producing their devices for patients with retinal dystrophy. And all of those devices were either epiretinal, so implanted on the surface of the retina, or subretinal. And the reality is that the surgery was much more complicated. It had a lot more risks associated with it. And hence, they've not continued to produce those devices. We believe actually that the simplicity of our surgical approach and the stability of the device makes it a really good, long-term option for patients with RP.
And how long do you anticipate that it will last? Like, when would it need to be replaced at all, or is it so stable that you don't really foresee that?
Well, the two patients that have had it in for 10 years, the devices are very stable in the suprachoroidal space. We have done some preliminary lab work about removing the devices and replacing them, and that's certainly surgically feasible, but obviously it's not ideal. The better option is to be able to upgrade the software, the vision processing algorithms, and alter, the hardware, the vision processing units, and that is a much better option for patients and participants.
We have a question here from someone who wants to know from you, Penny, are you able to comment on the work Elon Musk is doing regarding a brain implant for vision?
I can comment on, there have been a couple of groups around the world that have done cortical implants, small in number of participants in trials. And I think that, interestingly, this is an option for essentially people who've lost vision due to optic nerve disease or severe trauma. So there's always been an interest from the American military, for example, in these devices because of severe eye trauma during war. With a device that goes within the eye, the optic nerve needs to be functional to transmit the visual impulses back to the cortex. So it is an option for people that have severe diabetic retinopathy or glaucoma. And, some of the early work on these cortical devices is promising.
That's great. Kylie has asked, are you taking DNA samples from the West Australian data bank to use as well, or is it separate?
A great question, Kylie. So we work really well and really closely with the guys in Perth. And so the way that it works is if we have a clinical trial that's coming up or a particular study that we're looking for, we can actually contact their registry and say, these are the criteria, this is what we're looking for. Can you please send out an email? So we don't have access to any of the data on the Perth registry, so I can't go through there and look at the genes themselves, but I can ask them to advertise the work that we're doing. The other thing that we do is science, if we involve someone in our venture registry and we're trying to learn more about their gene and they say, oh, I've actually given a sample to Perth already. Once we have consent from you, we can then go back to Perth and say what your findings that data as well. We need to know that it's very strict privacy rules, so I don't get any of the data from Perth unless you consent to that, but we can.
That's great. So somebody's asked the same question: is the genetic testing you perform different to the WA registry? But I think we've just answered that. Really
Yeah,
well.
definitely. I guess, and I might just add as well. So there's kind of, we work very well, you know, in parallel and entwined. But it's definitely different ways that we do things. So we tend to do what's called a panel, first of all. So there's there's over 300 different genes that we know can cause inherited retinal diseases. So we'll take a saliva sample or a blood sample and just test for those genes. So that's sort of almost like the screening starting kind of test. What we then do from a research point of view, if we can't find the answer, we can then offer our participants the opportunity to go to the clinics. So for example, the ocular genetics clinic at the Ayton Hospital, and they can do further testing. So that's when you get into the whole testing the whole genome and trying to find out, you know, the more hard to solve ones. So Perth probably falls a little bit into that second category as well. So they're very good at really trying to nut out the challenging cases and working with some of the best labs around the world to try and solve challenging cases as well. So yes, we definitely overlap to some degree, but we definitely have strengths that we combine as well.
That's great. I had a question. You mentioned when you were talking about the different trials that are on at the moment, that the Stargardt's disease trial is an oral therapy. What is that? How does that work?
So this is a little bit of a challenging one. I'm actually not allowed to talk about the details of that one too much, I'm afraid. But basically, there are different oral medications underway at the moment. And the idea is that they either are an antioxidant. So one of the most common areas of research now is antioxidant therapies for the eye. So it's trying to protect the retina as a whole. The other class of sort of drug treatments that are really getting a lot of interest is trying to interact in the disease pathway. So gene therapy is trying to correct the gene right at the start so you don't get the follow-through problems. What you can also do is some of these oral medications, for example, which are trying to block the pathway. So trying to protect the photoreceptors by getting in the way of the disease in essence, really. But yeah, unfortunately, I can't go into too much detail because it's still very early.
Okay, well, that's really an interesting thing. I guess I'd only ever thought about, you know, the genetic sort of side of things, but it's great to hear that.
And I must say, I think this is why I love this field so much, because, you know, there's bionic eyes, there's gene therapies, there's oral medications. We're looking now at intravitreal injections. So these are injections that go in similar to when you have macular degeneration. So they're not requiring surgery, theatre and things like that. They can be done in an ophthalmologist's room. So those sort of treatments are starting to come forward as well. So there's going to be more and more options coming down the track.
Fantastic.
And Lauren, I just want to say, I think that one of the things that's really interesting is that, you know, there are more of these gene agnostic treatments being discussed because there are a number of these mutations that are really quite rare. And also there's, you know, 35% of people with inherited retinal disease that don't have an identified genetic cause. So being able to develop therapies that will help, that are not dependent on the genotype, are obviously really, really important.
Absolutely, absolutely. And it also ties back to what I was saying before about, I guess, the ecosystem that we work in as researchers. So the fact that there's a lot of people still working on the basic mechanisms of disease. So, you know, if you have an inherited retinal disease, what's actually happening and why are the cells dying and what sorts of chemicals are being released in your eye that's causing that damage, that actually opens the door to new treatments as well. So none of it can flourish without the other aspects of research.
Absolutely, yeah.
We've just got one more question quickly for the air. Do you know of any research results into how effective treatments are like microcurrent stimulation, acupuncture, herbs, and hyperbaric oxygen therapy?
Absolutely. So I am very happy if that person wants to email me, I can send some papers through. There's definitely, in particular, acupuncture had a lot of interest probably about 3-4 years ago, and including electrical acupuncture. So rather than using needles, it was actually direct electrical stimulation. There's also a lot of interest in electrical stimulation to the eye itself and whether or not that can help to protect the retinal cells. So there is interest in all of those sort of spaces, and they're really kind of working in parallel as well with some of the more drug-based treatments.
Fantastic. All right. Well, thank you very much. I'm sure our time is now up. So I want to say thank you very much to Penny and Lauren for making yourselves available and providing such interesting presentations. And I hope everybody's found the session informative. If you still have some questions, please don't hesitate to contact us, and we will make contact with Lauren and Penny and put you in touch with whoever can help. Now, when you leave the webinar, you'll be redirected to a very short survey, which we would really love it if you could fill in. It just helps us to get feedback about today's session and to think about what sessions you might like to have in the future. We've recorded this session and we'll be sending out that recording in the next day or so.
Can I just say thank you so much to you, Sally, and Julia on behalf of Retina Australia. It's a great opportunity for us to be able to let you know what the funding helps us do. And, you know, research doesn't get anywhere, unfortunately, without funding. And we're very grateful for the support from Retina Australia. And we're really grateful for the interest that everyone has demonstrated today in our work.
Well, it's tremendously important to all of our members, and these are the most popular sessions are to hear about where the work is being done and where their money is going when they make a donation. And it's tremendous that you're available to tell us about this important work. So thank you very much.
Thanks once
Right.
again.
Goodbye, everyone. Thank you.